Okay, this video is going to try and look at meiosis, and hopefully by the end of this little video you'll know the stages involved and how it introduces variety. Okay, so prophase 1. If you imagine, if I draw this now, I've got two pairs of chromosomes here. There's a maternal set, and there's a paternal set. So this is a pair here, this is a pair here. And during interphase, these will have doubled up, and we're now entered into prophase, so we can now see them as their distinct chromosomes. Now what happens during metaphase is they align, but rather than aligning in one big long line down the equator, they align in homologous pairs. So what that means is we're going to have our large red one here, and a small red one here, and a large blue one here, and a small blue one there. Now, as in mitosis, that will have been brought about by centrioles pulling out spindle fibres to cause them to align. Now, this is very different to mitosis. In mitosis, they'll have aligned one after the other. So you've had the red here, the blue here, the red here, and the blue here. In meiosis, in metaphase 1, they align in these homologous pairs like this. Now, I'm not going to talk about it right now, but towards the end of the video I will. This is the really important stage here. This is where the variation in the gametes that are formed at the end of this split occur. Now, I'll talk about that towards the end of the video, but for now we're just running through the stages. So, in Anaphase, what's going to happen is the chromosomes are going to split as before, but they're now in their pairs rather than the centromere splitting. The spindle fibres will have been doing this ratchet action and pulling them towards the poles here. And then once we end up in telophase, they've reached the centrioles, which are here and here. So what you end up with will be two cells each with 1/2 of the pairs of the chromosomes. Although they've actually got the same volume of DNA, or the same amount of DNA, here and here, as in a mitotic split, they've only got half the genetic information because they've only got 1/2 of the pair of this chromosome and 1/2 of the pair of this chromosome. The fact is they've got 2 copies of that half and 2 copies of this half here. That's probably the hardest bit to get your head around. What we're going to do now is just concentrate on this split here, because there's a second split in meiosis. This is going to split down into 2 further cells here and here. This will split down into 2 further cells here and here. But just for simplicity, we're just going to concentrate on this half here. Okay, so prophase 2. The chromosomes are going to condense again, but it's just what we end up with at the end of meiosis 1. And then the centrioles are going to release spindle fibres. What that's going to do is cause the chromosomes to align along the equator. So now it's looking very similar to mitosis, but the centrioles, spindle fibres, aligning the chromosomes along the equator. These spindle fibres now contract. What that's going to do is break those centromeres. So the join between the two chromatids is now broken, now referred to as being chromosomes, and they're being pulled back towards the centrioles. Once you're in telophase, the chromosomes have now reached the poles. What you'd end up with are two, in this case, identical cells, but with half the number of chromosomes. Because if you remember, the parent cell that these came from will have looked something more like that. In this case, you've got half the chromosomes here and here, rather than a complete pair. Okay, so earlier on I mentioned the variation that's brought about in the gametes, and this all happens in Metaphase 1. Now there's two parts that you need to be aware of in terms of Metaphase 1 that produces the variation. This is the first one here, independent segregation. This is down to the way in which the chromosomes align in those homologous pairs down the equator. Now I've got two possible situations here. Situation 1 here, situation 2 here. Now this is just with two chromosomes, or two pairs of chromosomes I should say, 4 chromosomes in total. In situation 1 we've got both blue chromosomes on the left and both pinks on the right. After meiosis 1 you're going to end up with two cells like this. And as they split down into their two gametes during meiosis 2, you're going to end up with one of each blue chromosomes in each of these two gametes, and then one of each pink chromosomes in each of these two gametes. If the chromosomes are aligned just slightly differently along the equator, so this time you've got blue and pink, and then pink and blue, something very different happens. This cell here has got a pink and a blue, this cell here has got a pink and a blue, and as we go down those two splits there, what you end up with are two cells here, a blue and a pink, and two cells here, the blue and a pink. I've just tried to be simple there in terms of the colours, but hopefully you can appreciate just the way in which the pairs of chromosomes align can lead to variation. That term there is independent segregation. Now this is just with two pairs of chromosomes. In humans we've got 46 chromosomes, 23 pairs, and you can imagine all the different possible combinations. If we'd have got 23 pairs running down our equator, there's in actual fact 8 million possible combinations. So there you can see the gametes that are formed in humans, the eggs and the sperms. There's just 8 million, over 8 million possible combinations just by independent segregation. Okay, the second way during Metaphase 1 in which variation can be brought about is by something called genetic recombination or crossing over. So during this phase, meiosis 1, we had our chromosomes aligned in pairs, like so. Now, what happens is something called crossing over. So if you can imagine, rather than them aligning like that, side by side, instead this chromosome here is as normal. The outer one on the red will be as normal. But what happens is this blue, something like this happens, and this red, something like this happens. What you end up with is a crossing over here of the DNA strands. Now this crossing over point here is called a chiasmata. Now this can happen multiple times all the way along the chromosome. They cross at complementary places, but it can happen at multiple times. And what you end up with are, if I just draw it on here, in the centre, what you end up with is this chromosome is going to be the same. The outer one here is going to be the same, but what we end up with here is a blue one with a bit of a red tail there, and then the red one here with a bit of a blue tail there. So we've now got 2 unique chromosomes here. Now this can happen, like I say, multiple times along this pair of chromosomes, and then along all 23 chromosomes as well. So what this does is bring about huge variation, and that's why 2 gametes, the chance of them being identical to one another are incredibly slim, because you've got the independent segregation, the way that they align along the equator, and also this genetic recombination. And where and how these chiasmata occur, these crossing between the DNA strands occur, can be completely random. The final way in which genetic variation is brought about in organisms is just the very nature of gametes fusing. That's a random process, and a human male releases somewhere in the region of 300 million sperm every time he ejaculates. Which one of those sperm goes on to fertilise the egg, which one does it, is going to be leading to huge differences in the offspring, because each one of those sperm is going to be very different due to the fact that genetic recombination has occurred and independent segregation has occurred, creating those unique gametes with half the number of chromosomes in them. So there's 3 things: random fusion of gametes, independent segregation of the homologous pairs of chromosomes, and genetic recombination by crossing over, bring about huge genetic variation in offspring.